pleasant evening to all our dear surgeons here in Singapore, and a good day to everyone around the globe. I'm Eileen Padilla from Osteoporosis International, and I will be your moderator for tonight's session. Before we start, I want to thank Singapore Dental Association, Nuffield Academy, and Osteopore Academy for making this webinar possible. This webinar is about new biomaterials in dental implant surgery. Before we start, allow me to mention our housekeeping for tonight. The webinar is being recorded. You will get the recording after the event. Tonight's webinar is scheduled to last 2 hours, including our Q&A session. To be able to get the CDE points, you will need to stay tuned. All participants are muted to enable the speaker to present without interactions. You may submit your questions through our Q&A chat box, and we will answer them at the end of the session. Lastly, fill up the survey after the webinar. For SDA member, kindly include your DCR number for your CDE points accreditation, and for overseas surgeon, you will receive your certificate of attendance. Our program will be in 3 parts. The first part is about Regenerative Implant Technology to be presented by Dr. Lim, and the second part is about new biomaterials in dental implant surgery to be presented by Dr. Kumar, and lastly, our Q&A session. Now, I will briefly introduce Osteopore International. Osteopore is a Singaporean company strategically poised to be a leading medical device company in the region. We are involved in designing, developing, and marketing bioresorbable polymer implants for neurosurgical, orthopedic, and maxillofacial use. We are first in the world to receive US FDA clearance for 3D printed bone devices for use in craniofacial reconstruction. We are certified to ISO 13485 Quality Management System. In the year 2020, we reached the milestone of 50,000 implants. Other than our FDA approval in US, we have CE mark in UE, TGA in Australia, and HSA in Singapore. We are committed to innovation through international collaboration. Here is a short video clip. Let's watch this. Thank you for watching. Now, our first speaker is Dr. Lim Jing. He's the Chief Technology Officer of Osteopore International. Dr. Lim holds a PhD from Nanyang Technological University, Singapore. Prior to joining Osteopore, Dr. Lim conducted research on biomaterials for tissue engineering and regenerative medicine and developed material fabrication platforms. Dr. Lim published 14 articles in internationally peer-reviewed journal in that time. Dr. Lim joined Osteopore in December 2014, and he has led Osteopore to important regulatory and quality milestones. In addition, Dr. Lim led the expansion of the product and therapy portfolio and contributed to the improvement in the manufacturing efficiency. Let's welcome Dr. Lim Jing. Dr. Lim, please.
Hi, Aline. Thank you very much for the introduction and a very good evening to all our participants for today. It's with great pleasure that I am able to share a little bit of our regenerative technology with you. So before I get started, perhaps if I could get the permission to share the screen. Okay, so yeah, I believe you can see my screen right now. So I'll go ahead and start. So what I plan on sharing with everyone this evening is about Osteopore's regenerative implant technology. And again, just to introduce myself, I was a mechanical engineer and also a bioengineer. And I do have a strong interest in translating tissue engineering concepts into clinical application. A lot of it stems from my previous research and close workings with clinicians in various animal study settings. And I got the dream of being able to realize these concepts in clinical practice at Osteopore International. So perhaps a little bit about the history of the company. Somewhere in 1992, the first fused deposition modeling printer, or called an FDM printer, was commercialized by a company called Stratasys. I think we may be aware of this company right now.
Hi, Jing. Sorry, just to, could you share your camera?
No, I can't.
1 second. Could you try now? Yeah.
Yep. Okay, so I'll carry on. So I was saying in 1992, we had the first commercially available FDM printer, or 3D printer, back then it's called. And 4 years later, the company's founding scientists and clinicians got their hands on 3D printers to start working on the research. And about 7 years later, the company was formed. Somewhere in 2006, we received our FDA 510K clearance for craniofacial applications of bone void fillers. And that was a landmark approval for us. In 2007, that was followed by our ISO 13485 certification, along with HSA's voluntary medical device regulations. In 2009, we were first awarded the CE mark of conformity. So that allows us access into European countries that follow the European conformity standards. From that point on, we continued to develop applications and innovations. And in 2016, we moved into our current facility, which is a purpose-built MedTech facility at Jalan Tukang, just very nearby Jurong Point. In 2017, we had a landmark case, this time in Australia, for regenerating 36 centimeters of the shin bone in an Australian patient who had unfortunately lost his original shin bone due to a spontaneous onset of osteomyelitis. In 2017, we started our first in human for rhinoplasty. That was done in Korea, and that also led to resounding success for us in that space. In 2019, the company became a listed entity in Australia. So as you have seen earlier, we are a Singapore medical device company. The scope of our activities include manufacturing, design and development. In Singapore, we do direct sales and marketing of our devices. In 2021, we reached a milestone of 60,000 implants globally, and we have also key approvals outside of the US, including Europe, Australia, Taiwan, ASEAN, and India. We are committed to innovation through international collaboration. The concept of our technology follows that of tissue engineering and regenerative medicine. So on the left, on the green donut circle, you will see a combination of growth factors, biomaterial scaffolds, and cells, which we can culture in vitro and generate what we call an engineered tissue. In the truest and purest concept of tissue engineering, such an engineered tissue would be generated ex vivo and then be implanted into the patient. However, due to significant regulatory hurdles, this remains to be an application. Conversely, we have thought of a different approach to applying tissue engineering concepts in a clinical setting, and that is a sub-branch of tissue engineering known as in situ tissue engineering. To achieve this, we would require a good three-dimensional scaffold that would be able to house autologously derived stem cells and growth factors in order to derive what we call a regenerative implant. So this is the centerpiece of our technology, and all the products that are designed are then based on this concept. 3D printing has many applications in the medical device industry nowadays, from models for surgical planning to surgical guides, and even to permanent implants. A lot of companies and even practitioners have taken to 3D printing with a lot of interest and applied them in various clinical settings. For Osteopore, what we believe is to charter the new future, which is to generate regenerative implants through a bioresorbable scaffold fabricated with a biomimetic structure that allows for tissue and vascular ingrowth. So this short video at the bottom right shows a simple process of 3D printing quality control checks, and that is conducted at our GMP facility at Jalan Tukang. This shows how our scaffolds enable tissue regeneration. So at the very beginning, there would be blood vessel that will go through a reparation phase. And what you see in the images as slightly yellowish tinge, that's original bone. And whatever that is in white struts would be the scaffolds. So as the blood vessels are repaired, there would be an extensive fibrin network that forms along with, and eventually we get revascularization. Due to the unique structure that we have created, we are able to see good penetration of vessels into the scaffold, not just around. And because of the intimate contact between scaffold and bone, this also encourages osteoblasts to migrate onto the scaffold and eventually proliferate and start laying down bone-like ECM to facilitate tissue regeneration. Over this period of time, the scaffold will slowly be absorbed and degraded in the body, eventually allowing the patient to recover with their own bone tissue. So what is the material that we use? For us, we strongly believe in using a slow degrading material known as polycaprolactone. So if we need a material reference, polycaprolactone is readily found in monocryl sutures, as highlighted here. Monocryl sutures are in fact a copolymer of glycolide and caprolactone. In our case, we are 100% polycaprolactone. This is a widely read and reviewed article that was published by one of our co-founders. It is to highlight the rise and interest of polycaprolactone in the 21st century, and also trying to explain why the rapid release of acid byproducts is not ideal, as it may lead to local inflammation. These were actually reported in bioresorbable device literature, and hence it also shaped their concept and philosophy around the ideal biomaterial to facilitate tissue regeneration. So what we believe that would go in tandem with the bone healing process is a material that gently degrades over a longer period of time. The resorption mechanism for polycaprolactone is hydrolysis. Its byproducts are gentle and can be metabolized into carbon dioxide and water. So on the right bottom chart here, we would see in black the bone healing process and the osteopore scaffold degradation process. You will notice that the scaffold stays in place for longer than necessary. And that again is to match the philosophy of our co-founders, which is to ensure that there is no excessive production of acid byproducts in order not to affect the healing process. As a result of this philosophy and technology, we have been able to develop multiple products across the cranio-maxillofacial region. Broadly speaking, we will split them into patient-specific implants and standard implants. In the neurosurgical space, we have patient-specific cranioplasty devices, as well as standard implants for burr hole reconstruction, whether with or without a catheter or shunt, a craniotomy gap filler, as well as an osteomesh for cranial vault remodeling. This mesh has also been used in pediatric cases of cranial synostosis. As we move downwards, we go to the orbital region. We have made customized orbital reconstruction devices that have led to very encouraging bone formation and patient functional outcomes. Standard implants would be also the osteomesh that is pliable, malleable, and conforms to the orbital floor. We move into the oromaxillofacial space, and for customized solutions, we have customized mandible or chin implants that will augment or reconstruct the lower body of the mandible. In addition, we have been developing our standard implants for socket preservation and guided bone regeneration applications, as we will share today. So as a Singaporean company, we are proud to say that we have a global footprint based out of a HQ in Singapore. We are present in many countries and many regions, and we subscribe to the most stringent standards and regulations that are required for medical devices. Between 2015 and 2021, we aggressively grew our product portfolio from 4 products to 223 products, including customization. From here on, I would like to explain a little bit about how did we get to this point. I would like to give a snapshot of Osteopore's progress from pre-2000s until the present day. From pre-2000 to 2005, I will qualify this period as the period where we were sowing the seeds. We spent 7 years in R&D, as I've explained earlier. We published more than 50 publications on in vitro and in vivo preclinical studies. We also invested in clinical trials to assess the safety and effectiveness of our devices in bone reconstruction applications. From 2006 to 2010, we began to grow the body of clinical evidence that we have. And that led to us being able to secure our regulatory approvals, notably from the US FDA, CE marking, as well as HSA. During this period of time, we did not rest on our laurels, and we continued to develop our technology and clinical portion. From 2011 to 2015, we began to align our resources for commercialization preparation. During this period, we have published more than 10 clinical publications, as well as collected 10-year retrospective review of our clinical data in a single institution. We managed to publish 3 of these articles recently. And from 2016 until now, we have focused on our organizational sustainable growth by strengthening our team with talent. We have clinicians that are on board with us, and we continue to commit to clinical education through the conduct of webinars and workshops. Last but not least, as we are very focused on innovation, we would like to continue this innovation by working hand in hand with clinicians in order to bring ideas into reality. So broadly speaking, the innovation process starts with identifying clinical unmet needs, the conception of an idea. We will proceed with certain amounts of bench and animal testing, proof of concept, and naturally protecting the intellectual property of the parties involved. Subsequently, the development phase is typically long and arduous, but is systematic and methodological. We follow the standards written in the ISO 13485 in terms of design and development. And this waterfall model here is a very typical process that is also used by the FDA's quality system regulation. At each step of the way, we ensure that we have verified our design and validated our design against the user's needs, and also conducted sufficient risk assessment and risk benefit analysis. After we have conducted the development process, we are now ready to submit our products to obtain market access. And to do that, we need to get regulatory approvals from the countries and jurisdictions that we are interested in. That could be the US or CE marking. But the other portion of bringing a product to the market is also education and gaining acceptance from our users. Beyond that phase, we move into the post-market realm, where we have to continually collect clinical data, publish them, continue training with our surgeons, and also as part of this process, perhaps innovate and find new products that we can work on together. So at this point, innovation again leads back to the source and the beginning, and we continue to run through the cycles of innovation to commercialization. So from the early days, we have conducted our clinical trials, clinical testing, and we have published these findings in publications as well. We have followed that up with 3 10-year retrospective reviews, as I've mentioned earlier, and these were published in the recent years. The next section, I would like to explore and show how we derive at the osteoplug for alveolar ridge preservation. So this device is meant to plug, support, and regenerate the alveolar ridge after the tooth has been extracted. The design of the scaffold is conical in nature and is easily shaped or trimmed in order to fit snugly into the socket. So the first phase of this procedure typically begins with a tooth removal that can be done as atraumatically as possible. Without the tooth, the jawbone may recede, and the geometry and the height and width may be lost as a result. The current practice is to graft bone, and our device is also meant to achieve this. After the device has been put in place, we will cover it with a membrane, and then we will achieve primary closure. The next phase is the reparation phase, which we have seen earlier, so I'll skip to the next portion, where a dental implant is prepared to be placed into the socket. Followed by the crowning process. Over the healing process, the bone will eventually turn into the patient's own, and there will be no more foreign material in the patient. So how did we get to this point? We invested in a clinical study led by Dr. Gobitin from National Dental Center to study the effects of this device when used for ridge preservation. So in this study, 13 patients were randomized to the test of control groups. Alveolar ridge height and width measurements were made at baseline and 6 months post-extraction. We also conducted CT scans for trephined bone in order to determine whether or not the device has successfully regenerated bone in the dental socket. So going through a little bit on the surgical handling, a suggested method of using this device would be to raise the trapezoidal buccal mucoperiosteal flap. followed by atraumatic tooth extraction. The device can be cut and shaved to fit snugly at the crestal half and the 2/3 aspect. Finally, we will incise the periosteum of the buccal flap to achieve tension-free closure with a 4/0 Vicro. This is an image of the healed tooth at about 6 months and also preparing the site for dental implantation. With the trephine bone that we managed to extract from the patient, we conducted a histology using a tricolor stain, as indicated here, as well as a von Cossa stain. We found that the tissue consisted mainly of mineralized lamellar bone within the porosities of the PCL scaffold, and that is directly opposed to the struts of the scaffold. We found a network of bony trabecula, and that is very typical of cancellous bone structure. Interestingly and importantly, we found that blood vessels were also observed, and this corroborates with our initial research findings where the scaffold structure was able to facilitate tissue and vascular ingrowth. There were few inflammatory cells and no signs of fibrous encapsulation of the PCL scaffold. We also demonstrated ability to maintain the ridge height and width. The key findings were that the PCL scaffolds were clinically immobile within the socket with an ISQ of above 65. There was ingrowth of relatively hard mature bone. There was less change in ridge height compared to the control. And there was less vertical resorption in all three aspects: mesial buccal, mid buccal, and distal buccal. Next, I would like to present a case study that was published in Dental Asia, and I would like to credit these case details and images to Prof. Raymond Wong from National University Center for Oral Health. The background of this patient is as such. It's a female patient with a failing dental implant at tooth 1-2. The clinical examination and radiographic findings revealed that the implant was subcrestally placed and resulted in significant bone loss around the implant. This is a mock-up of our device, and this shows the intraoperative handling, which we may trim with a pair of sharp surgical scissors. So the approach of Prof. Wong was to prevent future restoration issues and hygiene problems. In order to do that, they had to remove the implant and rebuild the bone. In order to achieve the vertical and horizontal bone regeneration aspects, particulate bone grafting was used in the socket area, and the osteomesh was then placed over. So the concept of this is illustrated in the figure on the right. So you have the alveolus bone highlighted in orange, the particulate bone graft that would fill this aspect here, and the osteomesh was then placed over. A membrane was placed over the osteomesh. So this is the intra-op picture of an impression of how the osteomesh would be placed in situ. The healing process from 6 weeks to 6 months and eventually to post-implant placement demonstrated that there was good bone maintenance, both in terms of the height and the width, and this led to successful implant placement subsequently. On the right, we have an image of the ridge at 10 months. So the 6-month follow-up radiographic image shows thin identifiable bone at the grafted site over the top of the alveolar ridge, and this indicates that the success of the surgical method for GBR of the alveolar bone. The surgeon also noted that implant placement was stable. This case did not proceed without any minor issues. So actually, there was a minor exposure at the extreme border, as you can see circled in white over here. And the method of handling such a concern would be to trim away using a pair of surgical scissors. It was notable to understand that there was no infection at the site, nor at this area here. So the learning points from this case was that the osteomesh is comparatively stronger as compared to collagen membranes, and hence it was able to provide a protection for the bone graft. Despite being exposed to the oral environment, the clinical experience from this case also indicated that it does not lead to infection and causes less inflammation to the mucosa. Before I end my presentation for today, I would like to demonstrate that we are committed to contributing to improving patient quality of life. On the left, this is a Vietnamese patient that suffered from spina bifida, and we sent our osteomesh over to this patient and donated to her so that she was able to cover that bone defect through the works of the neurosurgeon and was able to finally sit upright post-surgery. This second panel shows a 36 centimeter bone reconstruction at the tibia, which saved the patient's leg from amputation. I've met the patient in person 2 years after the reconstruction, and he was able to ambulate without crutches and get on with his normal life routine without much issues. This third case is actually a Vietnamese patient that was treated in Singapore for extensive craniofacial defects that required reconstruction. Again, in this case, we contributed our mesh for the neurosurgeon and plastic surgeons to successfully reconstruct her face, and this will be just one of the many reconstruction surgeries that are required. The fourth case is that of a bone cancer patient. She's called Asher. She had 15 centimeters of her tibia to be removed, and that resulted in a substantial gap in the tibia. Using a particular technique developed by a plastic surgeon, we were able to reconstruct 15 centimeters of her bone, and now she is also able to ambulate with the aid of crutches as she continues her recovery process. For this last patient, is an Australian patient, but he suffered an accident in Vietnam. And I would like to share with you the video of his recovery process.
Hi, Jing. There's comments that there's no sound from the video.
Okay, just let me try and get the video in.
Thank you.
Man has
Are you able to hear it now?
Yes.
received a 3D printed skull implant. Brodie Ellis underwent a groundbreaking 11-hour procedure carried out by a team of about a dozen surgeons, doctors, nurses and technicians. And now, just a few months later, he is finally back home with his family. It's been a life-saving surgery for the 26-year-old who was left with a stage 4 brain injury following a motorcycle crash in Vietnam back in 2018. Earlier, I spoke with Brodie and Dr Michael Vargles.
It was a really long road out of hospital, and there's a whole new appreciation for life after what I went through.
Wow, incredible. And your family, I know everyone was with you. Dr Vargles, though, what you have done and your team is truly incredible. I know this isn't your first time working with 3D technology, but just explain to those of us that don't have medical knowledge how you use 3D printing to fix Brodie's skull.
So as you know, Brodie was missing quite a large section of skull. He'd already had a reconstruction of that skull using fairly standard materials, plastic essentially, which had been produced with 3D printing as well. The way the materials are used and implanted in the body, which was quite unique in Brodie's case. As you know, we'd had some experience with this in the past in a leg, reconstructing a leg bone. Brodie and I had a chat about how that might be applied in his situation. Being the courageous person that he is, he was well up for it. So once the 3D printed implant is manufactured and it's made of a material that essentially dissolves and disappears, we need to make sure that in its place bone will form, and Brodie's own bone, more to the point. So we've harvested a piece of tissue from his knee and then auto-transplanted it up to his scalp. So he's now sporting this lovely lump over here, which is actually skin from his knee, and underneath that is the bone lining, which produces new bone. This is the first time this has been done in a skull. We did a scan about 6 weeks after the operation, and even at that stage, you can see that there's new bone forming both on the outside and the inside of the 3D implant. So, yeah, the process has already started well and truly.
Brodie, you must be in awe of everything that's happened. You've been through so much. I know this has been an incredibly long, tough ordeal. You had to have your leg amputated, but just tell me, I know there was something specific, wasn't there, that you asked Dr. Vargles before you lost the lower half of your leg?
I asked Dr. Vargles if he would be able to skin graft my tattoo from my left foot onto my right foot, and he told me he was doing a favour by getting rid of it.
I love it that you can maintain such a great sense of humour. What are you doing next? What's your next big hurdle?
One step at a time, but I hope to be studying again by the end of the year in the summer semester.
Good luck. It is amazing. I have no doubt that you will achieve all that you set your mind to. You're amazing. Brodie, lovely to talk to you. And Michael, congratulations to you and your team. It's incredible work. Thank you.
Okay, so before I end my presentation, again, I would like to just share that we are really very keen on collaborating to develop new products and technologies that would, in the end, benefit patients and also improve the healthcare standards for everyone. So thank you very much, everyone, for your time this evening, and I'd like to pass it on to the next speaker.